So that's objective number 1 covered. We've related the structures, properties, and functions of water in life. Now let's look at objective number 2. Predict what will happen to cells in hypotonic, isotonic, and hypertonic solutions. Now, in order to be able to do that, we need to visit a concept called diffusion. Diffusion is the movement of a substance from a high concentration to a lower concentration across a concentration gradient. Now, it's kind of common sense. Everybody knows that if you're sitting in class and there's 25 people and the guy in the middle, well, lets a little fart out. Who's going to smell it first and who's going to smell it the strongest? Well, you know that the closer you are to him or her, the more intense the smell is going to be. And that as that fart cloud moves away from itself by diffusion, area of high concentration to an area of low concentration, it gets weaker and weaker and weaker until it diffuses away completely. Now, water's a little bit different. So much so, in fact, that we have a unique word to describe the diffusion of water: osmosis. So osmosis is the diffusion of water. It's the movement of water from a high concentration to a low concentration across a concentration gradient, but it's a little bit different. Why is that? Well, let's not forget, each of the water molecules is hydrogen bonded to the next. So the way that it moves, it's not going to be able to move like a gas in every single direction. Instead, it's going to basically move in a continuous flow. Okay? So what does this mean in terms of cells? So consider these two containers. In container A, we have some water molecules spread out. And in container B, we have some water molecules, and we're going to add some salt molecules in between. Now, these salt molecules are going to be attracted to the water because of its polar covalent bond, and water, we know, is a universal solvent from the previous objective. So these salt molecules are going to dissolve into the water, and they're going to take up some space. And what that's going to do is that's going to push the water molecules further away from each other. So what we can see is that by adding a solute to water, we're actually decreasing the concentration of the water molecules themselves. And what that means is the more salt or solvent that we add to water, the lower the concentration of the water. Now, words like hypertonic, hypotonic, and isotonic can be broken down. Remember, biology is a language, and it's better to break these things down. So let's look first of all at what does tonic mean. Well, tonic or tonicity is a description of the amount of salt dissolved. Okay? So hyper means a lot. Hypertonic means a lot of salt is dissolved. If we have a lot of salt in the water, that means all the water molecules are going to be pushed apart. So a hypertonic solution has a lower concentration of water. How about the word hypo for hypotonic? Well, hypo means weak or less. So hypotonic means we have less salt dissolved. Now, what does that mean? Well, if there's less salt dissolved, that means the water molecules are going to be closer together. There's less pushing them apart. And so we're going to have a higher concentration of water in a hypotonic solution. What about iso? Well, iso means equal. So isotonic would be equal amounts of salt. What does that mean? Well, look, all three of these terms are comparative terms. Comparative terms only make sense when you're comparing two things. For example, if I said I am bigger, it doesn't make any sense. But if I say I am bigger than a mouse, it makes sense. Okay, I might be bigger than a mouse, but I'm not bigger than an elephant. So I am bigger on its own makes no sense. The terms isotonic, hypertonic, and hypotonic are comparative terms. So when we describe something as being hypertonic, meaning it has more salt than something else, we're saying it has more salt. As a biologist, we need to remember the more salt it has, the less water it has. So a hypertonic solution is simply a solution that has less water and more salt than another solution. Hypotonic then therefore would mean that we have less salt and more water than another solution. And of course, isotonic would imply we have equal amounts of salt and so an equal concentration of water. So if we are to achieve objective number 2, we're going to need to be able to predict what happens to cells in these different types of solutions. Well, let's look first at an animal cell. So an animal cell is basically a cell with just a cell membrane. It's a lot like a balloon. Here we see an animal cell placed inside a solution that is hypertonic. Let's remember what that means. It means the surrounding solution has a higher concentration of salt, therefore a lower concentration of water than the cell. We know that osmosis is the movement or diffusion of water from a higher to a lower concentration, so that means the water is going to leave the cell, and the cell is going to shrink. So we describe the cell as being shriveled. Now what happens if we place an animal cell into an isotonic solution? Well, isotonic means equal levels of salt, so therefore equal concentrations of water, which means the amount of water moving into and out of the cell is equal. So what happens to a cell in an isotonic solution? Well, absolutely nothing. What about a hypotonic solution? We place a cell, an animal cell, into a hypotonic solution. What we're saying is that the surrounding solution has less salt and therefore a higher concentration of water. So water is going to move from the higher concentration into the lower concentration inside the cell, and the cell, just like a balloon, is going to swell up and eventually it's going to burst, which we call lysing of the cell. Now that's not going to be the same for a plant cell. A plant cell has a cell wall. That means it has its own container. So what happens to a plant cell inside a hypertonic solution? Well, it's not going to shrivel away completely because the cell wall is going to maintain the cell's shape. Instead, it's going to be what we call plasmolyzed. The inside part of the cell is going to strip away from the cell wall. In an isotonic solution, a plant cell wall is described as being flaccid. Okay? What does that mean? Well, it means the cell itself isn't pumped full of water. Now, for an animal cell, that was a good thing. For a plant cell, it's not. If you've ever seen a plant that hasn't been watered in a while and it starts to droop, that's because the cells are flaccid. They're not full of water. So for a plant cell, unlike an animal cell which wants to be in an isotonic solution, a plant cell would prefer to be in a hypotonic solution. Okay, we put the animal cell in a hypotonic solution and it burst, but the plant cell will not. Why not? Well, instead, it's going to absorb a lot of water and it's going to push on its cell wall. It can't burst because it's contained inside the cell wall. We describe this as being turgid, and this is actually the state that the plant wants the most. So plant cells prefer to be in hypotonic solutions for this reason. So that's objective number 2, being able to predict what happens to cells in hypertonic, hypotonic and isotonic solutions. Now, why is that so important? Well, that allows us to know how water is going to move around plants, animals and cells in general. And that's really important for things like hydration. It could also save your life. Consider the story of Douglas Robertson. Douglas Robertson and his family went for a cruise around Jamaica, and unfortunately they were shipwrecked. Adrift at sea for many weeks, of course they had a number of worries. What were they going to eat? What were they going to drink while they waited to be rescued? Well, turns out eating wasn't a problem. They were experienced fishermen and they had all the necessary equipment to go out and catch fish, sea turtles, and lots of other tasty things. Their problem was water. Now, being adrift on an ocean, you might think, well, why don't you just drink the salt water? Let's remember that salt water, seawater, is hypertonic compared to human cells. And so if you were to ingest the hypertonic salt water as it was inside your stomach, rather than giving you water, it would actually absorb the water from your body. We know that osmosis is the movement of water from a high concentration to a lower concentration. Salt water has a lower concentration of water due to its higher concentration of salt. So drinking the salt water would actually have made them more thirsty and killed them quicker. Now, Douglas Robertson's mother was a nurse, and she had a good knowledge of biology which saved all of their lives. She knew they needed water, they needed hydration. And the only water available that wasn't hypertonic was the rancid water mixed with fish blood and sweat and all sorts of other nasty stuff that I can't even say on the bottom of the ocean. If they were on the bottom of the boat. If they were to drink this, well, they would get very sick and probably die. So how can you ingest it? Well, she got a syringe from the first aid kit and she fashioned an enema. And for those of you who don't know what an enema is, well, there are two routes to get something into the body. There's the front door and there's the back door. And an enema goes in the back door. And so what she did was she filled up a tube full of all of the dirty, rancid water on the bottom of the boat that would have made them sick had they drank it, and this water was funneled into the Robertson family's back doors. From there, it was able to move by osmosis into the cells of their body, providing them with some hydration. Was it pleasant? I'm sure it wasn't. Did it save their lives? I'm sure it did. Why did they tell that story? I guess they had to explain where they got the fresh water from. So, biology could save your life.